Thanks, Ben. Can you see my slides?
Yeah, we can see your slides and we can hear you.
Okay. So hi, everyone. My name is Wendy Wong. So I have been analyzing other people's genome for research in the last 10 years. And today I'm going to share with you how I analyze my own family's genome using data from direct-to-consumer companies. So this is the brief outline. So I'm going to introduce you to human genetics and also tell you a little bit about the differences between these direct-to-consumer genetic testing companies. And then I'll go through some examples of my own analysis in my family's data. So this is a very brief introduction. So we human as a multicellular organism have cells, and then each cell have consists of DNA, and most of us have 46 chromosomes and 22 pairs of autosomes. So we get 22 from our mom and 22 from our dad. And then we also get an X from our mom and X or Y from our dad. And then together there are 3.2 billion of base pairs, so those are ACTGs, and then they encode for roughly 20,000 of the protein encoding genes. At the DNA level, you and I are very similar, so we are in fact over 99% identical. But these small variations explain a lot of things, our different in looks, height, obesity, and how we react to certain drugs and our risk to certain diseases. So here I'm showing one of the differences in our genome. So assuming the human genome reference is a G in this position, and then a person with 2 Gs, so getting one from each parent, we call it homozygous reference. And then the next person has an A and a G, so we call it heterozygous, and then we denote it as 1 because it's got one of the alternative allele, and 2 if it's got 2 alternative allele, and this become important later. Recently, there are so many different direct-to-consumer genetic testing companies. The most famous ones are 23andMe and Ancestry DNA, and they both use genotyping technology, and I'll get into that later. Denti Labs and Nebula Genomics are doing sequencing, which used to be super expensive. The first human genome project cost $1 billion, and now you can get it for $299 from Denti Labs. But what can you actually learn from DTC genetic testing? So a few years back, my family, so my parents and myself did 23andMe. And then recently we also got our genome back from Denti Labs. So there's two difference in the two companies offering. 23andMe offers genotyping. So if you imagine our genome is like a book, you're basically 23andMe are looking at these different just a portion of the genome and at these sites, and these sites are picked very carefully, and usually they represent the variation between individuals. And 23andMe looks at roughly 600,000 to 1 million of these variants, and the raw file I get from them when compressed is roughly 8 megabytes. So Denti Labs offer sequencing. So in theory, you're looking at every single site of the 3.2 billion bases, and you try to chop the human genome into pieces, and then try to map it back to the human genome, and also we try to cover each site with at least 30 reads. So the raw file I get from Denti Labs is roughly 50 GB, and it contains 700 million of these reads, and each read is 150 base pair long. and roughly 70% of them map to the human genome. And I'll get back to that later. And with that, we can call variants. So small variants are the ones that 23andMe are looking at. So particular sites or small, like maybe 2 nucleotide or up to 10 nucleotides long. And then most of us have roughly 3.5 million of these that are different from the reference. And then with sequencing, we can also look at copy number variants and structural variants, which I'm not going to get into today. So you can actually learn a lot from your germline DNA. I'm going to cover some of them today. I'm not going to talk about estimating age, but you can actually do that because telomere length decreases with age, and loss of Y chromosome in male also decreases with age, so you can use that to estimate your biological age. And also, you can also estimate the HLA genotypes from DNA. So this is important when you do a transplant, and also HLA is responsible for the immune system response. And also you can get the blood type as well. So one of the common things that most these companies provide is Ancestry. And so 23andMe and Ancestry.com both provides that. And here I'm doing my own analysis using the data that I got. So to do that, you need a good reference data to so that you know what each ancestry looks like. So I'm using a public data called 1,000 Genomes, and they sequence 2,500 individuals from all over the world, from the 5 superpopulations, so European, East Asia, mixed Americans, South Asia, and Africa. And so you get a variant file that looks like this. So each line is a variant, and then these columns are each individual in the project. So for example, this individual has a reference allele and the alternative allele, and this person has 2 alternative allele. So if you count the number of these alleles, you get a matrix like this. So I added my genome, my family's genome, and encode it like this, and then feed it to PCA, and I get a plot like this. So as you can see, my family is right in the middle of the East Asia cluster, which sounds about right. So this is pretty easy, but 23andMe can do a lot more because it has a much bigger reference. So they can actually go down to the regional level, and it tells me that I'm from southern China, which is accurate as well. And next, I want to go into single gene diseases. So these are the diseases that are caused by one gene. So like this person, he has many variants. He has also cystic fibrosis, and the reason that we think he has it is because he has these 2 variants that are damaging mutations in this CFTR gene. In my own case, I know that my dad and I both have G6PD deficiency, and I just don't know why. So this disease is interesting. So we're not supposed to take certain drugs or food. Like I was told that I'm not supposed to eat lima beans. But there's one advantage of G6PD deficiency is that most people who have it are resistant to malaria. That's why it's more popular in malaria regions, like some part of Africa and part of southern China. And 23andMe only looks at those variants that are popular in the African population. So it tells me that no, you don't have any variant, which is not right. So I took the data from DentiLabs, which sequenced the entire gene, and I imported that variant data into Google BigQuery, and then I joined it with a annotation database by Tut Genomics, which has 8 billion variants, and then I only look for those ones that are damaging. So to score greater than 0.9. And then I found the variant. So this is a very damaging missense mutation that would make the protein non-functional in the G6PD gene, and my dad and I both have it. And so I'm more fortunate than my dad that I have 2 copies of X. So I have 1 good copy, but I probably won't be eating lima beans anytime soon. So let's look at the complex traits. So these are the diseases that are caused by many genes. So each gene have a tiny effect on the disease. So and together they cause the disease. And there are a lot of traits, not necessarily disease, are also multigenic. So for example, 23andMe can predict your bitter taste, finger to length ratio, saturated fat from the polygenic variants. So to calculate your own risk, there's a formula. So this beta value is the contribution of each variant to the disease, which we get from association studies, and you multiply this with number of that variant in the individual, and then summing them up, and then you get the polygenic risk score. So here I'm using a R package called bigrquery, which allows me to interact with bigrquery. And bigrquery has 2 interfaces. One is the low-level DPI interface that you directly feed it with SQL code, or you can use the dplyr code, which is a lot cleaner and nicer. So here I get a table connection to the table that I created, which I join my family's data with the beta values for many different traits. And this is what the table looked like. And then now I'm looking at a trait called drive faster than motorway speed limit. So this is a study that I'm using the UK Biobank data with 500,000 individuals. And then I can do the standard dplyr code, mutate to get the product, and then I group by the sample name, and then I summarize and then get the PR score for my family. And here, this in the middle individual is my mom, who has the lower score, who's less likely to drive faster than the motorway speed limit, which is probably true. So I also did the same thing with the 1,000 genome data. So this is 2,500 public data available, and I did the same thing, and I plotted stratified by the superpopulation, and we can see that this green shade here is East Asian, who are less likely to drive faster than the speed limit, and the blue is the Europeans, who are more likely to do so. And this is very preliminary data. I haven't done extensive QC, but it's just fun to look at some of that. And I want to point out that this is a big data set, and the running the job on Google bigrquery is 2 seconds. And although it's not super cheap, the Google bigrquery charges by the number of the size of data is through, so it's $5 per terabyte, and the first terabyte is free. So next, this thing is more useful. It's called pharmacogenomics. It tells you your reaction to certain drugs. So there are 2 famous examples. One is CYP2C9, which determines your sensitivity to warfarin, which is a blood thinning drug. So it could be potentially life-threatening if you have one of the mutations and got prescribed the drug. And also CYP2D6, which controls the metabolism of codeine, which is a painkiller. So there are many research tools that can predict the genotypes of these genes, and I use LD, which is super convenient. So you can just do a pip install, and then point it to the sequence file, and then I get the results back. So these 4 are the public genomes, and then these 3 are my family. So my dad and I are both normal, and my mom has the star 36 allele fusioned with a star 10 allele. And I did tell her to be more careful when she's prescribed those drugs and probably get the proper clinical testing when the doctor prescribed her those drugs, just to on the safe side. So lastly, I also want to show you that what you can do, what else can you learn from your saliva. So so when you send your saliva for DNA testing, they're looking at all the DNA in saliva. So most of them did come from you, so they come from the buccal epithelial cells and your white blood cells. But it basically is sequencing everything, so including bacteria and virus. And in fact, when I aligned it to everything, I found out that my parents just had orange before they provided the sample. So so again, you took the sequence file, and then I obtained the unaligned reads. So these are the reads that are not aligned to human. And then I feed it to Kraken 2, which is a tool that can do classify metagenomic data, and then taking the report and feed it to Pathian metagenomics data browser. And this is a very nice data browser that's powered by RShiny, and you get many nice plots, and this is one of them, and it shows the relative abundance of the microorganisms in my saliva. And then if you click on one of them, it shows the number of reads that are aligned to that particular species compared with the other 2 samples. These are my parents. And I also noticed that my parents' oral microbiomes are a lot more similar than mine to them, but I don't have a good sense of what is normal, so I will have to dig into that. So to end this talk, I want to give you a list of tools and platforms that I use. And also, if you are interested in learning more, you can go to Bioconductor, which has 1,800 R packages for genomics. And there are some courses and computational biology books that you might find it interesting. And thank you for your attention.
Thank you so much, Wendy. We do have one question, and it's about big data. Do you have any advice for somebody handling big data in R?
So there's RSpark if you want to do it that way, but there's traditionally that's not what R is good for, and that's why you see a lot of packages that are working with, say, the BAM file, which is 50 GB. They're using Python or C++, and R is better on handling the summary data. But right now there's a Sparkler and RSpark that could basically leverage Spark to handle big data.
Fantastic. Thank you so much.
Thank you, Ben.